Yes, sir. I'm Director of the Office of Biotechnology Activities at the National Institutes of Health. My office is located within the Office of the Director of the NIH. I report directly to Dr. Lana Skirball. Thank you, Mr. Chairman and members of the subcommittee. As you've heard, I'm Amy Patterson, Director of the Office of Biotechnology Activities at NIH. And I'm honored for the opportunity to be here today to discuss NIH's role and activities related to the oversight of gene transfer research. And I wish to extend the agency's deep condolences to Mr. Gelsinger and his family. Mr. Chairman, I'd first like to say that NIH understands that our ability to support and conduct clinical research is predicated on trust. We have a profound compact with patients and their families to ensure that every measure humanly possible is taken to ensure that those trials are conducted in the safest and most ethical manner possible. We do this through a comprehensive system of federal laws, regulations, and guidelines that are designed to protect human subjects in clinical trials. Jesse's death is a harsh reminder that investigators must comply with these standards and principles for the safe and ethical conduct of clinical trials. As you've already said, gene therapy is a relatively recent and still experimental application of recombinant DNA technology. It's caught the public's attention, not only because of its promise, but also because of the deep ethical and societal implications that research which involves the basic unit of human life, DNA, holds. Gene therapy may be used to provide a copy of a normal gene, to directly alter or repair a mutated gene, or to regulate the expression of other genes. Clinical gene transfer research has grown exponentially since the first experiment was done back in 1989. To date, more than 4,000 patients have participated in clinical trials, and over 400 trials have been performed. The vast majority of these studies are designed to assess safety and toxicity. Only 1% of the trials have progressed to clinical efficacy studies. Because of this, we feel it's more accurate to call this area of research gene transfer rather than gene therapy. Gene transfer trials target a wide range of disease, including cancer, HIV infection, cystic fibrosis, as we've heard this morning, cardiovascular disease, arthritis, and hemophilia. NIH currently supports approximately 160 gene transfer trials in a wide array of disorders. NIH's unique role in the oversight of gene transfer research reflects the public's longstanding and special concerns about this area of research and NIH's leadership in scientific innovation. NIH's stewardship over this arena of research is accomplished primarily through three tools: the NIH guidelines involving research using recombinant DNA molecules, the Recombinant DNA Advisory Committee, otherwise known as the RAC, and public dissemination of data and information about clinical gene transfer trials. The NIH guidelines clearly set forth standards and principles for the conduct of human gene transfer trials. They also articulate the role of the RAC and the requirements for the submission of protocols and adverse event reports to NIH. The RAC has several critical roles in the oversight of this arena of research. It recommends changes in the NIH guidelines to reflect advances in the knowledge of the safety and science of this field. It conducts in-depth public review of all novel gene transfer protocols. And through national policy conferences, it explores in depth the very complex scientific, social, and ethical issues that this technology has raised as it's progressed forward. These conferences have yielded very important consensus recommendations that guide the field this day. The NIH guidelines clearly, unequivocally state that investigators must report all serious adverse events to NIH immediately. Each and every time an investigator submits a protocol to NIH, they receive a letter that restates their obligation to fulfill this requirement. The purpose of this reporting is not for day-to-day individual monitoring or to place clinical trials on hold if need be. That is one function of the FDA. Rather, it is so that NIH can mobilize the RAC and scientific community to bring to bear their expertise and experience to better understand the potential implications for adverse events and what those implications may be for ongoing trials, as well as to help design better trials in the future. In addition, we can initiate data collection for a comprehensive and public review by RAC and ad hoc experts as necessary. And as you know, this is exactly what we did one day after being informed of Jesse Gelsinger's death. Reporting of serious adverse events also allows us, in the long term, to recognize previously unrecognized trends that may have significant implications for the safety of patients enrolled in these trials. And this entire process fosters a broad public awareness on which public trust and confidence in this field is predicated. We recently became aware that there was widespread noncompliance with NIH's requirements for reporting adverse events. We're actively analyzing the extent of this noncompliance and the possible contributing factors for this noncompliance. Investigators have violated the federal guidelines. Our top priority is to ensure 100% compliance, and we've taken 4 immediate actions. First, we directed institutions and investigators involved in gene transfer research to ensure and report back to us about their processes for ensuring at their local institutions full compliance with the NIH requirements. In addition, we asked them to report any adverse events which had not been previously reported as required to NIH. Second, we've established, in collaboration with our colleagues at FDA, a new process whereby we know each time a sponsor submits an adverse event report to FDA. It's important to note that this is a backup system. It allows us, if we haven't received the same report, to follow up with clinical investigators. It in no way obviates the obligation of the investigators to report those same events directly to NIH. Third, we're actively exploring additional ways to enhance our reporting requirements, and we're making sure that all pertinent information regarding the safe and ethical conduct of these trials is reported in a timely fashion to the RAC and to the scientific community and the public. This being done with full protection of patient confidentiality by the stripping of individually identifiable patient information. Fourth, the NIH is going to be making a series of site visits to NIH-funded institutions. The purpose of these visits will be to ensure compliance with the NIH guidelines and to examine compliance with other NIH policies, including financial conflicts of interest. One of NIH's primary oversight responsibilities is ensuring public access to information in gene transfer research. This information has always been publicly available. At each RAC meeting, we describe any new gene transfer trials, any new adverse events reported since the last meeting, and new developments in the field. This data is up on NIH's website. Every gene transfer protocol, in addition, is available to the public upon request. We're in the process of developing a technically advanced interactive database. This database is designed to serve a variety of users, patients, the general public, as well as the scientific community. And it will enable people to search for specific variables, aggregate data, query, answer their specific questions about this field of research. And finally, in order to be sure that our oversight role is as optimal as it can be, because that's what we owe patients, an advisory group is evaluating NIH's oversight of gene transfer research, and the NIH director has specifically asked this group to develop and consider several options, among which is whether approval authority should be restored to the RAC. In conclusion, gene transfer clinical research holds tremendous promise to treat, cure, or even prevent a wide variety of human diseases and conditions. And if we're to realize this promise, investigators, funding institutions, and regulatory agencies must work together to ensure that this research is conducted in a safe and ethical manner. If we're to improve the nation's health through research, we must all work, as this meeting here today is one important step forward in assuring, work in concert to protect the most important participant in clinical research endeavors. That's the patient. Mr. Chairman, members of the subcommittee, this concludes my statement. NIH appreciates this opportunity to participate in this critical hearing, and I'm pleased to answer any questions. 160 NIH-funded or supported clinical trials. NIH has received over the 10 years that gene transfer research has been underway about 370 protocols. We classify them a bit differently than FDA. As Dr. Siegel explained, an IND may encompass several clinical trials using the same product for a variety of clinical indications. We tend to classify each trial as a separate entity. There may be different principal investigators in charge of each clinical trial. Right. There are, again, over the decade of history, we've had 372 trials. At present, there are 160 actively funded NIH trials. There's a difference in the way NIH counts or tallies trials compared with FDA. As Dr. Siegel said, an IND, an investigational new drug application, may encompass a number of trials. We see all trials that are conducted either at an institution that receives any monies for NIH monies for recombinant DNA research. So anybody who's doing molecular biology, we see their trial. And that certainly includes the smaller subset of NIH-funded trials. And I'd have to call on my colleagues at FDA to answer that question. Right. First, I'd like to set the record straight, if I may, sir. The 691 serious adverse events that were reported encompass, because NIH guidelines require any serious adverse event, whether it's related or unrelated to the protocol, expected or unexpected, if an adverse event occurs in a patient while they are enrolled on a clinical protocol, the current guideline requires that event must be reported immediately. So in our preliminary analysis of this 691 events, most of these events are by and large due to the patient's underlying disease. As you probably are well aware, the majority of gene transfer trials are conducted in patients with severe diseases, such as advanced malignancies. And the majority of the events that we have received reports of indicate the progression of a patient's underlying disease, a side effect of a medication such as chemotherapy. We are in the process of conducting an in-depth analysis of those adverse events in collaboration with our colleagues at FDA. And to date, we have seen no evidence that a heretofore unrecognized problem in gene transfer research is among those reports. But nonetheless, we take very seriously the duty of looking through those reports and making sure. When the 691 adverse events were reported, those events were reported in response to an NIH call to all investigators in the field using adenoviral vectors, the same vector that was used in the trial at the University of Pennsylvania. That call for specific safety and toxicity data was issued on October 1st, and those reports came in October and November of last year. Right, and it's also important to remember that those 691 adverse events represent a 7-year cumulative history of the use of adenovirus. They are not events that occurred in 1999, over a 7-year period. The first adenoviral trial was performed in 93. No, sir, the guideline says immediately. That's not stated any more specifically. And that is one of the things that we're clarifying. What is the scope of the reporting and what are the timelines? But the current guideline reads, investigators must report any serious adverse event immediately. Yes, sir. Again, the 691 adverse events are the whole universe of adverse events that happened in adenoviral trials. They are not, and that is a different requirement than the FDA requirement, which is expected, unexpected, and related to, or possibly related to, the intervention for immediate or expedited reporting. The 39 events referred to in Dr. Kirstein's letter are those events that were reported to NIH immediately, in other words, in the time frame that we require. That's not to say that many of the other 652 adverse events, we had seen previously many of them, but they were not reported in a timely fashion. So I would like to set the record straight that it is not that NIH didn't see the other 652 or a subset of them. It is that they were not reported in a timely fashion to the NIH. I think it's important to realize that many of those adverse events had been previously reported. They may not have been reported immediately as required. I think that the death of Jesse Gelsinger galvanized the scientific community to bring to bear its experience and expertise so that we could fully understand any lessons that Jesse's death holds for the use of adenovirus vectors in particular and for the field in general. The NIH guidelines are quite clear in their statement that investigators must report. And again, we send every time we have contact with investigators when they submit a protocol of their obligations. We are undertaking now an extensive analysis to look into the extent of the noncompliance. Well, first let me say that we find the current noncompliance not acceptable, and we will do everything we can to address it and work with the subcommittee. That being said, to answer your question, the letter that investigators receive is a personally addressed letter that says quite clearly a whole paragraph is devoted to the obligation, the responsibility of investigators to comply with the standards for the safe and ethical conduct of gene transfer research, and in particular, it unequivocally states their obligation to report serious adverse events to the NIH. And again, we will be looking very closely into the extent of noncompliance and taking every step to make sure that there is 100% compliance. No, the NIH guidelines is a lengthy document. It applies to a wide variety of recombinant DNA research. Appendix M is the chapter that directly addresses human gene transfer clinical research. Appendix M is just a few pages long. The responsibility of institutions, the institutional review boards, and the investigators, a triad at the local level for ensuring compliance and reporting, is recapitulated throughout the beginning of the NIH guidelines in sections 1, 2, 3, and 4, and I'd be happy to supply you with the exact citations. And again, as I just mentioned, each and every time an investigator submits a protocol, we remind them in no uncertain terms of their obligation to report. I think that investigators have violated the federal guidelines. That's not to say that we will redouble our efforts and take every additional measure. I should say they should know that. About 93. The other trials, and let me correct you, yeah. 93 of the 372 trials that NIH has, so adenovirus vectors account for about 25 to 30% of all current trials.
All right.
In 1999, NIH received approximately 100 reports of serious adverse events, approximately 70 of which were on other types of trials. The NIH has, in the RAC, has consistently looked at the serious adverse events reported and discussed them at RAC meetings and made this data available to the public, and carefully monitored what is going on with the other types of vectors that are used. Well, again, I think that we're taking every step we can. We've contacted directly all institutions conducting this research to ask them to report to us any heretofore unreported serious adverse events. Yes, that's correct. We have had a number of adverse events reported in response to that letter as well. I can tell you directly now, I said we got 100 reports in 1999. We received reports after the November letter of approximately 40 adverse events. There were deaths that were reported that were attributed to the progression of underlying disease or concomitant medications. There were none that were attributed directly to the gene transfer. Well, this is a question that we've been asking ourselves, and while we do, as we've been discussing at length here this morning, believe that the guidelines and our letters sent to investigators are quite clear, it's obvious that there has been widespread noncompliance with the NIH guidelines reporting requirements, and that's unacceptable. The NIH director has formed a special advisory group that is looking at the role of NIH oversight in this field of research, and has specifically asked this group to address the very question that you've asked, sir. What options need to be considered, including a possible return to RAC's approval authority. Right. And again, we've asked ourselves this same question. Let me also, just for the record, RAC relinquished approval authority on October 31st, 1997. But we're in collaboration with FDA conducting a comparative analysis of the numbers of adverse events reported to NIH and FDA, as Dr. Siegel's already mentioned. And that analysis is focusing both on the time period prior to the relinquishment of RAC approval and to the period after RAC approval was relinquished, so that we can try to get at the question you've just asked. The preliminary analysis that we've done of 34 INDs does not indicate a change, a dramatic change, but I think that their sampling errors, which protocols you pick, has a lot to bear on that question. Yes, the informed consent of patients is a absolutely critical component of clinical research, and the RAC has a long-standing history of carefully evaluating not simply the informed consent document, but the entire process by which patients are informed, notified of an existence of a protocol, talked to about their potential participation, how risks are presented to them, and these recommendations on specific protocols have always been forwarded from the RAC to the local IRBs for their consideration as they are looking at these protocols on their desk on a day-to-day basis. I think that because the informed consent process is it's at the heart of the matter, as you've said, that we can never be satisfied with the process. We've heard this morning very good suggestions on how the process could be improved. I think that the NIH has in the past and is committed to continuing and redoubling our efforts to communicate at the local level, participate in meetings with IRBs. So while I think we have a good process in place, I think that there is room for improvement. Yes, there's a working group of the RAC that is specifically charged with undertaking the revision of that proposal in light of the public comments that were received. The position of the RAC to date has been that adverse event reports belong in the public domain so that they can inform other investigators, they can inform prospective patients. And the RAC has held to that principle. It's a complex issue because some of the manufacturing details, issues of patentable information about how a product, some specific detail of manufacturing, perhaps need not be included in an adverse event. And there's no interest on the part of NIH to include such information if it has no bearing on the adverse event and it's an accurate interpretation. Regarding the second point, patient confidentiality is a critical issue as well. And while it's important that the information be out there, it's important that it not contain information that could identify specific individuals. And I think those two principles the RAC has endorsed. I think what is at issue and what the group is trying to balance is the universe of adverse events that are reported. Do we report every single adverse event immediately, or do we report serious, potentially, possibly related to the intervention immediately, and then all others in an annual report? I think that is the part of the puzzle that the RAC is grappling with now, and at their upcoming meeting in March, they will present on their deliberations, and in conjunction with the advisory committee to the NIH director, they will be preparing a final recommendation on how NIH will receive, analyze, and publicly disclose this critical information.